All the cells in the human body, including primary spermatocytes in men and primary oocytes in women, are diploid, which means all of them have 23 pairs of chromosomes. If 23 pair of chromosomes are taken out and sequenced for the number of base pairs they contain, it will be about 3.2 billion base pairs. So we can say that the human genome contains 3.2 billion base pairs. What's the human genome? Or for that matter, what's the term genome mean? The genome implies all the genes of a particular organism in one copy. This includes the genome of mitochondria and chloroplasts as well. And when we say all the genes, we also imply those non-coding segments. In other words, all the coding and non-coding segments that contain in 23 pairs of chromosomes and in mitochondria are included in the genome. For each species, there is a definite set of genome. Genome refers to the total genetic content of a cell, of a diploid cell, of an organism. Now, what is the epigenome? Epigenome refers to all those molecules that are intimately associated with the genome. All those molecules primarily mean histones and non-histone chromosomal proteins, but then there are small molecules also. They are associated with them to affect the activity and inactivity of genes. For example, there may be a few small molecules that may bind to promoter and enhancer to activate, accelerate the activity of a gene. By activation of a gene, we imply the transcription of RNA from the gene. So histones, non-histone, and other proteins that regulate the activity of genes constitute the epigenome. Let me give you an example how they affect or activate genes. You see, the histones are supposed to be general suppressors in that they bind the DNA or gene in such a manner as to bring about superfolding of the DNA, forming what is known as chromatin. The chromatin is highly contorted super three-dimensional structure. Now, in that state, when the DNA is in highly contorted fashion, there cannot be any exposure or accessibility of the codes contained in the DNA. The codes must be available to be seen, to be recognized by RNA polymerase to read out and to form complementary sequence that's called RNA. So the codes must be available. The availability of code for transcription implies activity of the gene. If the codes are inaccessible, then of course the gene is said to be inactive. And this is done by epigenetic elements. How do they do? Is a very complicated phenomenon. Let us suppose then that the histones are actually basic proteins that are associated with the DNA because of electrostatic interaction. As you know that the DNA is negatively charged macromolecules. So they are associated with positive charges contained in histone proteins through their basic amino acids. These interactions result in contortion, inaccessibility of the codes. If these histones are somehow removed or actually slightly made loose, they can, of course, inactivate the gene. How is it done? If histones are activated upon by enzymes that acetylate them, phosphorylate them, or methylate them, then there will be some kind of change in the three-dimensional structure of histones, as a result of which they may dissociate temporarily from the DNA they have bound to. So these structural modifications that are brought about by enzymes in histones cause activation, inactivation of genes depending upon what they are actually doing.